The development of a new drug costs, on average, $5 billion. This is why many of the most innovative ideas for treating or even curing diseases will never get to be tested in humans. It simply costs too much. Only a handful of new drugs can enter clinical development every year. Why is it so expensive? It is not the cost of new advanced technologies for molecular research that sucks up all this money. What makes the average cost of drug development so prohibitive is the very large and very long process of testing drugs in patients. Many of the drugs that are tested work on only a small group of patients. These experimental drugs may have a large effect on a small group of patients or have a small effect on many patients. To see these effects, you will need to test the drugs on thousands of patients over a long period of time, years, monitor them closely, and compare the patients that got the drug to other patients. Such clinical trials eat up more than 90% of the direct cost of a single drug. If we could identify the group of patients that will respond to the drug before the clinical trials, we could have established the statistical significance of the effect of the drug faster with fewer patients. This will not only make the clinical trial much cheaper, it may also lead to fewer failures. Obviously, being able to match patients to drugs will affect not only the success and the cost of drug development, it will also improve treatment. It will allow physicians to choose from among the available therapies the one or the combination that will work on a given patient at a given time with less side effects. The main tool to do this is biomarkers. A biomarker is something that can be measured and is associated statistically with the biological state. For example, elevated levels of a prostate-specific protein is a biomarker for prostate cancer. Or high blood pressure is a biomarker for being at risk of heart disease. Biomarkers may be diagnostics, helping identify patients with a specific condition, or they can be prognostic, identifying patients that will have a specific condition or will respond to a certain treatment. For example, the presence of a certain mutation or a combination of mutations may be diagnostic, telling us that the patient suffers from a certain condition. Alternatively, specific mutations in a tumor cancer can be prognostic, telling us that the patient will respond to a certain treatment. Using biomarkers, of course, is an old clinical concept. However, genomics, proteomics, and big data allow scientists to identify increasingly complex biomarkers. Here's an example. In many types of cancers, some patients respond well to treatment, while others do not. Conventional clinical measures such as age, background, or even stage and grade of the cancer often provide poor prediction of which patient is going to respond well. We've seen that molecular technologies allow us today to measure accurately which gene is expressed in every moment in a single cell. Such data can be obtained by old technologies like microarrays or newer technologies like RNA-seq. So these technologies were applied to cancer cells from patients. Then computational analysis of the profile of expression of hundreds of genes revealed significant differences in gene expression between patients that have a good chance of responding to treatment and patients that did not respond well to treatment. Note, there was not one gene whose expression level was significantly different between the two groups. It was the complex pattern of thousands of data points that revealed the difference. This figure shows one of the earlier studies where gene expression patterns were able to differentiate between patients with estrogen receptor plus, namely patients with cancer that respond to estrogen, shown on the left, and estrogen receptor minus, namely patients whose cancer do not respond to estrogen, which are shown on the right. This classification between estrogen receptor positive and negative has prognostic and therapeutic consequences. Based on findings like this, the FDA approved diagnostic tests that measure the level of expression of many genes and use them to direct treatment based on the predicted prognosis. Researchers are now mining large sources of data with sophisticated algorithms to find new biomarkers. Biomarkers don't have to be only molecular or biochemical. As we mentioned in the previous unit, it has been shown that computational analysis of medical records of children can identify, for example, children at risk of child abuse years before health professionals raised concern. Genomic variations, medical records, proteomic analysis, gene expressions are only some of the sources of data that are used today to identify new biomarkers. They increasingly allow physicians to select the most appropriate care for a given individual. They may also revolutionize drug development, lowering costs and leading to precision drugs that work for specific people.